my lab is interested in using natural products as potential therapies for neovascular eye diseases, specifically wet age-related macular degeneration, but also retinopathy of prematurity. And we discovered in the literature a very interesting chemical compound that was isolated from a member of the orchid family by a group in Korea several years ago. And they had shown that this naturally derived product had some effects on blocking the growth of blood vessel cells, and in animal models of retinopathy of prematurity and wet AMD, or at least choroidal neovascularization. So we set out to synthesize this compound from scratch from chemical starting materials rather than extract it from a plant as had been previously done. So I collaborated with a synthetic chemist named Seong-Yong Seo at Gashon University in South Korea to produce the compound, which he was able to do. And then we brought this newly synthesized molecule that had never been made by human hands before to our lab, where we tested it for its anti-angiogenic activity. And we showed that the compound that we had made did indeed have the ability to block the growth of retinal endothelial cells. We then went on to make some structural derivatives of this compound, so trying to change the structure, keeping the same sort of parent compound, but tweaking it so that it might have better activity, potentially down the road better properties as a drug-like molecule. And so we screened somewhere around 60 different molecules. Many of them my collaborator Dr. Seo made. Some of them were obtained through a collaboration with Dulcie Mulholland, who works at the University of Surrey in the United Kingdom, and she extracts these compounds from plants. So some of them were newly extracted from plants, and a few of them we were able to buy from commercial sources. We found that some of these compounds had substantially more potent activity than the compound that we had originally synthesized, and also showed some selectivity for endothelial cells, so blood vessel forming cells, over other ocular cell types that we tested.
What were your animal models and how did you use them?
So we actually haven't used animal models yet in our study. The natural product, as was previously isolated and previously reported by others, was tested in a mouse model of retinopathy of prematurity, which is very similar to the human disease, in which neonatal mice are exposed to high levels of oxygen after birth, and then are brought back to normal oxygen levels, and they get aberrant retinal endothelial cell growth. The choroidal neovascularization model that was used previously involves burning the choroid with lasers, which again causes aberrant choroidal endothelial cell growth. So we plan to move our compounds into these animal models in the near future.
So tell us about the results of this particular study and the next steps that you envision.
Certainly. So we're particularly excited about the novel compounds that we've created that seem to have better activity than the originally synthesized natural product. And as I said, we will definitely be testing those in animal models in the near future to confirm that the activity that we see in cell culture does indeed hold up in animals, and that there are no unforeseen toxic side effects. From there, it becomes a matter of improving these compounds further, really tweaking the structures and optimizing their activity in the hope of eventually developing new drugs. And of course, for these diseases, there are effective drugs, but they are antibody-based therapeutics generally. They're very expensive, and not all patients respond to them. So there's a real need and a niche for potential small molecule drugs that we hope in the future to be able to fill.
And these molecules will be bioavailable orally or intravenously or both?
That's an excellent question. At this stage, we don't know. We haven't done any kind of pharmacokinetics or pharmacodynamic studies to see how these molecules behave in the body. I would envisage most likely an intravitreal injection, as is done for the existing compound or existing biological pharmaceuticals on the market.
What is your prospective timeline for this work?
The conventional wisdom is it takes at least a decade from interesting discovery in the lab to potential drug being tested in humans. So I would go with the conventional wisdom and say it would probably be a decade before we are in a position to safely introduce this or a derivative into humans.
From a translational perspective, when do you think you would be recruiting people into a phase 1 clinical trial?
Again, we are still quite a substantial way from that point. Certainly we need to make sure that this compound works as expected in animals, extensive testing, optimization of delivery. So it will be several years before we're ready for human trials, but we're certainly working as quickly as we can to move in that direction.
Thank you very much, Stuart.
You're very welcome. Thank you.